Hi and welcome to my fourth video in the series about membranes. Today we're looking at active transport. So here are some examples where active transport is used. Absorbing glucose by the small intestine and in the kidney tubule cells, salt uptake by roots of plants, and amino acid uptake by fungi. So essentially what active transport actually is, it enables you to move chemicals and molecules. Instead of down a concentration gradient, it enables you to move it against it. So instead of facilitate diffusion or osmosis, which are passive, which require no energy, because they happen down a concentration gradient, what you're actively trying to do is you're trying to move something from where there's not a lot of it to a lot of it, and that therefore requires energy and ATP. And so what we have here is an example of active transport specifically with sodium ions, and so they require a carrier protein in order to do this. What you've got here is sodium ions moving from the intracellular fluid, so moving from the inside of the cell to out. So we can first of all establish that outside the cell there's a big concentration of sodium ions, and that therefore this is why ATP is required in order to move them, because it's moving against the gradient. And so what happens is the ATP is broken down and is broken down into ADP and a phosphate group, and energy is released, and that energy is used to change the shape of the carrier protein. That change of shape can therefore release the sodium ions to the outside of the cell, and hence that's active transport. So we've looked at moving objects like, or molecules like sodium ions and water. But what happens if you want to move something much larger across the membrane, something like a protein? And for that you'd use the following processes: endocytosis, exocytosis and penocytosis. We'll have a look at each of those in a second. So in endocytosis, a chemical like a protein might bind to the surface membrane, and the plasma membrane basically folds around that object or that molecule, and it forms a sealed environment inside there, and so you can take in a chemical like a protein into the cell, and again without that change in the biochemistry of the cell. Now endocytosis and penocytosis are synonyms, so they're very, very similar to one another, and so for an A-level qualification you probably wouldn't be expected to differentiate between the two. Now endocytosis is where you take in a substance, exocytosis is where it's released, hence EXO, like exit. And so they use this organelle called a vesicle, and a vesicle is essentially a lipid bilayer that's formed around molecules that need to be secreted or got rid of. What happens is, because those chemicals may actually cause damage to the cytoplasm or change the chemistry of the cytoplasm, they're contained within a lipid bilayer, and that's referred to as a vesicle. That vesicle will then fuse with the plasma membrane, and then it will enable the chemicals or the molecules to be secreted without ever coming into contact with the cytoplasm. So in summary, active transport is the movement of substances across a membrane via a carrier protein against the gradient, and therefore requiring ATP. So it's not passive, it's active. Exocytosis is the release of larger chemicals from the cell, so exo, get rid, like exit. Endo is absorption of larger chemicals. Both processes use an organelle called a vesicle, which is a membrane-bound organelle, and it's used to transport things either inside the cell or outside.